We've known that the cells are grow in their suggested medium. However, as time goes by, many formulations are tested and become more suitable for their requirements, which can cause a lot of inconvenience when preparing large numbers of different kinds of cell culture media. We started thinking the possibility of culturing different cells by using only one kind of basal medium. Therefore, OneMEDIUM developed. This special basal medium is able to sustain cell proliferation, maintain cellular bioactivity, and most importantly, keep their gene stability. It has also been tested on different cell lines and compared to their suggested basal medium, which are those measures used in cell culture works. In our tested results, it shows that cells can even grow at low serum environment. Therefore, not only did we test the cells at the recommended FBS concentration, we also compared the cell culture performance at low FBS concentrations with other reduced serum basal media brands. In this test, cells were cultured by their suggested medium, OneMEDIUM at low FBS concentration, and the other brands of reduced serum formulations. The tested cells are highly applicable by using OneMEDIUM. Comparing with other cell culture media, their morphology are similar. By observing their growth rates using doubling time, they are various, but still comparable with those of the original medium. The most important thing is that we've done STR analysis on those tested cells. The STR analysis can specifically identify cell genetic profiles and demonstrate the cultured cells are the same as those in their original medium. We have also tested cell bioactivities on different cell lines. On protein expression analysis, 9E10 cells were tested. This cell line can secrete anti-semic antibody. Comparing cell doubling time among the media, OneMEDIUM shows great performance on this cell line. It has relatively shorter cell doubling time, and their anti-semic production capability was comparable to their suggested media. Cultured cell surface marker expression analysis, we tested on THP1 cells. This cell line exhibits CD45 positive expression. We cultured this cell line on different cell culture media, including OneMEDIUM. And in OneMEDIUM, it shows a better performance on their cell doubling time comparing to other two reduced serum media. Their characterized surface marker CD45 staining were analyzed by flow cytometry. The result shows a clear single histogram on OneMEDIUM condition at CD45 positive ratio over 99%. The OneMEDIUM can also be used for human bone marrow mesenchymal stem cell culture, and the cell maintained better performance than their suggested medium. It remains good cell doubling time, which means users will be able to harvest more cells in primary cell culture to apply on future differentiation demands. Moreover, their characteristic surface markers are well preserved. One of the most important things of verifying the stem cell's quality is their capacity of differentiation. And OneMEDIUM cultured stem cells also shows good performance here on their adipocyte, osteoblast, and chondrocyte differentiation. After serial tests, we launched this product to serve as a good alternative for major used basal media. It can simplify cell culture medium preparation and maintain good cell growth environment quality. Through our test, OneMEDIUM can sustain cell proliferation and maintain gene stability, remain stable cell functions of tested cells, and exhibit good cell potential in mesenchymal stem cell cultivation. You can find more product testing information through this QR code.